Welcome to Neurosalience, the OHBM podcast.
Hello and welcome to Organization for Human Brain Mapping Neurosalience podcast season 4. I am Ömer Faruk Gülban, who is the podcast production team lead of this season, and I am working on this podcast as a volunteer in the communications committee of Organization for Human Brain Mapping. Today, we are going to chat with Peter Bandettini, our host, to see what awaits us in the next season. Hello, Peter. How are you?
Oh, I'm good. I'm looking forward to the next season.
Yeah, me too. I am excited for it. How was your summer? Could you rest well? Are you
Uh,
ready?
yeah, it was a great summer. It was a great summer. It was very rejuvenating. Lots of family activities, and yeah. But I feel refreshed, and finally the weather's getting a little bit cooler, and I'm looking forward to that. Uh, so yeah, planning on getting into this.
Yeah, good to hear. So then let's start to talk about the podcast. So I was wondering if you had any highlights from the last season that you want to share with us.
Oh yeah. So last season, which was the third season, had 20 interviews with really outstanding people. A lot of people who I knew, a lot of people who I didn't know. Some of the highlights. Let's see. I'm going to go, approximately in
Mhm.
chronological order, but one of the first episodes. This actually the second episode technically was with Prantik Kundu and Charles Lynch talking about multi-echo fMRI. And, that's a technique that's catching on, and it's super useful for eliminating non-bold fluctuations. And Charles Lynch actually wrote a couple of really good papers demonstrating how powerful this technique was for doing group studies and making inferences really efficiently. It's like you basically get, if anything, you get multiple time series for the price of one almost. And there's more advantages of that. You can actually, model the signal and extract the bold versus non-bold, but you still have bold artifacts. I didn't really learn this, but one thing to highlight is that there's still an ongoing struggle with eliminating things like respiration, and that's also bold. So that was we might even have more episodes with that. So another one, Brat Biswal was talking about his history. He even gave a talk at OHBM on the history of how resting state fMRI was sort of discovered, and it's kind of a cool story of a serendipity and just trying things. And so he was one of the first people within the framework of AFNI of just saying, hey, maybe I'll use a regressor taken from the data, and see what correlates. And it seems like an obvious thing to do now, but then nobody thought of it, and the rest is history. And I think I would like to this season talk a little bit more, like delve into resting state. I know I had talked to Maurizio Corbetta in a past episode, and I think there's more there. There's much more there, and it's ongoing research. So a really cool episode that I did learn something. I learned something every episode, but talking to Mikkel Ramat from the Weizmann Institute, she was talking about real-time feedback. And initially when people think about real-time feedback fMRI, they think, oh, well, you have your function fed back to you, and you're trying to change your activity in some way, and that kind of works. But she's putting forth a really, really clever idea, and that is it's covert. It's important to emphasize this here again, is that because I think it's really potentially powerful, and that is where subjects are viewing something and they don't really know what task they're doing, but the idea is that you're given a stimulus that every once in a while, like maybe fills in a little bit more, and or does something that causes some sort of dopamine response brain, some sort of pleasure response. And the idea is that what's happening is that the algorithm is watching the resting state fluctuations. And whenever there's a resting state fluctuation that shows correlation between the network they want to strengthen, they give this stimulus that gives dopamine. So the idea is that dopamine reinforces the strength of that connection. And it seems like it works pretty well for, she was looking at autistic subjects, trying to change the network connections. And, it's still, new stuff and it's preliminary, but it's exciting. And that gave me an insight in that, there's so many things that can be done covertly, that the algorithm is just watching your connectivity patterns. And before you know it, you have a different connectivity pattern. So there's been other work that was related to this, that not just by her, but that was cool. Let's see. So I had a wonderful talk with my old friend, Jeff Binder, who's always doing really insightful, clever work on understanding, he focuses on language, but he really has unique methods for just understanding how the brain is organized in general. And so that's an episode worth looking into. Also, Lily Mojica Parodi. She is a researcher who's probably a little bit less known, but she's doing really unique research on understanding the effects of ketogenic diets on brain function. And she has some pretty compelling data, both fMRI and otherwise, that shows that that if people start a ketogenic, have ketones instead of glucose as the energy source for their brain, it reduces a lot of the what she considers spurious fluctuations or noise in the brain. So that was cool, and it sort of ties into the idea that, for the past 75 years, people have been using ketogenic diets for people with epilepsy.
Mhm.
And so there's something going on there, and that and she sort of has compelling data that shows that. So that was new and cool. There were great talks with Todd Constable, who opened my mind to the idea that of prediction, using fMRI for prediction, and sort of along the lines of trying to look at individual subjects. And he has a good perspective on that. And also, he had some interesting recent data showing that, and this is something that's sort of like something that's in my head, and I can't get it quite out of my head. The idea that the problem that when you parcellate brains, everyone's brain is different, and so each individual parcellation is different, and so it's hard to normalize those. And also, he actually brought out the point that during the time series, the parcellations, the size of these nodes might change, and the tortuosity changes. So things are variable, and we kind of need to get our head around that to properly compare individual subjects. So I had a wonderful talk with Stephanie Forkel, who's been helping out with this podcast before, but she's doing great work looking at white matter, not white matter function, but functional activity as defined by the white matter tracks connecting the activity. So she's been mapping that out and making these disconnectivity maps that are really clinically useful. Audrey Fan was a person I interviewed, that because she is a physicist working really very much in the clinical realm, trying to use fMRI in a different way, more physiologic fMRI, looking at vascular reserve. And if anything, I think that might be one of the first clinical applications of fMRI, aside from, it's already being used a little bit for pre-surgical mapping, but we're always looking for biomarkers to use for prediction for psychiatric disorders. I think that's still kind of far off. But I think the more tractionable use of fMRI is extracting really subtle vascular physiology. And she's been sort of one of the pioneers of that. So I talked with Andreas Bartsch and Natalie Voets, who, along the lines of clinical applications, is their work. They wrote this huge white paper, like 120 pages or something, on all the ins and outs of properly doing pre-surgical mapping of language. And it opened my eyes to how difficult it actually is to bring a research technique into the clinic. And you really want to have standardized methods. You want to have something that a physician can rely on, and that's a very, very high bar. And generally the clinical realm is very conservative, so it's hard to really get those things in. And there's as they're writing the paper, actually, there's, even among people who really want this to be used clinically, there's some disagreement. I know Jeff Binder has some views on what's the best thing to do and what's not. And then Andreas has other views, and I think that this is a process of coming up with a consensus, and you just have to work and work and work at it and just pound away. Lastly, had a great episode talking with Sanja Kotz and Shella Kielotz. I'd like to talk to both of them about their own science, but they were editors of Neuroimage, and as everyone knows, there was a massive migration of all the editors from Neuroimage to Imaging Neuroscience. The main reason for that was Neuroimage did have open access, but they charged a fee that was high, and it sort of puts up a barrier for people who want to submit papers and have them open access. Because I think what they did was sort of a further catalyst for a process that's inevitable, and that is having all the journals transferring over to very sustainable open access. It's hard. I mean, there's still some costs, but it doesn't have to be what it is right now for many journals. So that's an inevitable process. That's just will continue. Yeah, and also lastly, I had a conversation with Russ Poljak, which we barely scratched the surface of everything he's been doing. So I'd like to have him back on. He agreed to be back on to talk more about what he's done.
Actually, we didn't have time to edit and publish Russ Poljak's episode. Therefore,
Okay.
I can say that he will be one of the first episodes in this season, so our listeners can look forward to it.
Okay, something to look forward to. And also, Andrew Jan, who does education at fMRI, he's also hasn't been put out. So those will be the first episodes
indeed.
of the fourth season, and then I'll be interviewing people. And to be honest, nothing is fully set yet, but I have all these people to invite in my queue, and I would welcome anyone listening, because the season's still wide open, to give me suggestions. If you want to be on, if you have something to talk about, if you have something to debate, or if you know of someone else that would want to be on, just give me an email. I'll share my email right here on the podcast. I'll share my Gmail account. It's just Bandettini, BANDETTINI.Peter@gmail.com. So give me an email. Right.
I hope you will not regret this decision, Peter. But let's see what interesting things will come out.
Yeah, definitely. All right. Well, that was pretty much my highlights. There were many more, but I figured.
Yeah, great. Really interesting and good to hear. Our listeners should go back to the last season and follow up on these topics if they are curious. So one last thing you didn't mention, maybe you forgot to mention, but we had a very, in a way, famous episode, or in our little bubble, I can say, and that was about a highly impactful paper that came out last year. It was about a method called Diana. And you might remember that you were very fast to get the authors to the podcast, and we were actually postponing all the other episodes and first editing that one to be really fast on publishing what authors think about this Diana paper. That was a science paper. And I was following how this paper was turning out, and recently I saw some actually cautionary notes about the paper saying, I mean, it's not a retraction or anything, but it was just saying that further experiments and testing validation might be required to trust whatever is written in this paper. Then I had this following dilemma, like should we hype up some new important papers and sometimes maybe feel that, oh, I need to drop everything that I do and switch to this new hype, new method, or should we be more careful and see how things turn out in the following weeks, months, years? And of course, the other part of the story is that as a source of distributing the scientific knowledge, we contributed to it as well as a podcast. And now maybe if something doesn't work out in the follow-ups of this paper, what would be our place as the Neurosalience podcast to
that's a good point.
Talk about these things?
Yeah. First of all, I purposely left out Diana just because we could spend some time talking on it here. So first of all, I'm generally I'm kind of an optimistic person, and very, when it came out, I also try to balance that with a certain amount of criticality. And, I was excited about it. I mean, I have to, full disclosure, I've been sort of trying to think about and work on direct neural current imaging for 20 years, from neural currents to other sources of contrast. And I haven't really been working on it much, just because of the fact that wouldn't be totally fair to assign a project to a graduate student or a postdoc, because it is kind of high risk. It's high gain, but it's high risk, and you may not get anything. But it's fun to think about, and every once in a while I try something. So Diana came along, and the paper itself was, I'm usually pretty critical of neural current or direct neural imaging papers, because they usually have some result that's barely detectable, and they're making something big out of it, and which is important, but it usually ends up not really being doable for a number of reasons. It's just too noisy, or you have to do it just right, or it's idiosyncratic, or whatever. And also, of course, all authors, including myself, sometimes fall into the trap of, you have to watch yourself. If you really, really, really want a result, you end up doing things that are biasing for the result, even without knowing it, even without being fully aware of that. So it's best to try to do your research with sort of this sense of like, okay, whatever the results are, they are. And once you start wanting a result, it's very easy to, even objective scientists, it's very easy to interpret the data in a context that makes it seem like it's convincing yourself that it's real, and then finding a good argument to convince everyone else. But the point is, I'm not saying that this is Diana. I read that paper, and they had like 4 different experiments that were, if they just had one, it probably wouldn't have gotten into science, but they had 4 different experiments that showed in different ways that this was a real effect. And not only that it was a real effect, that it was potentially even sensitive to membrane polarity, so it could be positive or negative. It could show suppression. So that was really cool. And they even showed layer Diana work, and it was just a tour de force. And so that was great. And also, full disclosure, I was after the episode, I started talking with Sanji Kim and Ravi Menon, and Sanji was in an awkward position because he's in charge of this group that sort of works with them to some degree. And he wasn't on the paper. He wasn't, there's no rivalry or anything like that. But he actually came to me and said, "Hey, there's some issues here." And so we wrote a paper outlining the potential issues. It's in bioRxiv right now, between Ravi and myself and Sanji and a few other people that were involved with the analysis. And the issue is this with Diana. And I actually don't know much, but all I know is that one issue is the potential. So I think that in the electrophysiology literature, and a lot of the researchers there come from that, it's quite a common practice to, if you have a non-performing animal, to sort of objectively measure that performance and say, oh, well, they're non-performing, let's take them out of the data. You don't average them together. And there's some thought, and I don't know completely what the truth is here, but there was some thought that, there was some selective averaging of not the animals, but potentially of the time series, saying, hey, this time series, every once in a while they respond, and every once in a while they don't respond. And maybe they were using the electrophysiology as an objective measure. I don't know. But the idea there, and so what the implications of that are, and so that's actually right. You have to be super careful when you select data. You have to do it, have a completely objective way of determining this, not using the data to select the data. But the implications, and also, I'm not saying anything that the authors haven't said themselves in public at ISMRM and OHBM, and that is. And so Ravi Menon did an experiment where he, and also Sanji, where, it was higher field, and they did many more averages, and they just averaged everything, and they saw nothing. And the authors were saying, well, that's because you used everything. And what we find is, if we keep on averaging everything, the signal goes away. And in statistics, that usually means it's not real. But their argument was that you have to be clever in how you select, otherwise, because the true neural response is variable. So we're in really really sort of treacherous ground here, and how to properly pre-select your data to average. And so that's where we are right now. And I would love to have Ravi and Sanji and maybe the authors of Diana on to discuss this further, because it really is important. And it is a paper that did cause a huge stir. I had a bunch of neuroscientists give me a call or just to email me saying, "What's up with this paper? Should I be doing this?" And here's my point. And also, we've been trying to do this in our group. We've been trying to reproduce the results in different way. We also, my thought is that, okay, let's just boost the signal noise as much as we can. Let's turn off the gradients. Let's just have a coil in the back of the head. Let's simultaneously measure EEG with humans at 7 Tesla and see what we see. And we see, and it's funny because we see things every once in a while that look like they could be compelling, but then we see stuff that's respiration artifact and who knows what. And so we're still not sure ourselves. But then what I tell these people is that, okay, this technique may or may not be real. I'm not 100% sure yet, but even if it is, the signal change, let's say it's real. Let's say it's totally real. They're really, really, really hard experiments to do. At least the way we conceive of it right now is that you have to have, the special pulse sequence. You have to have it super time locked. You have to have incredible control of the timing. And the percent signal change is an order of magnitude at least less than bold. And you have to also make sure that there's no bold seeping in here and there as well with your timing. So is it worth it? It might be in some contexts, some very, very specific. Let's say, yeah, if it is real, is it worth it? It might be worth it in very, very specific contexts, but it may not be something. I mean, bold was amazing when it came along because it just worked. It just worked. It was hard not to have it work, and that's why everyone uses bold, because it works. And sure, we would like more signal to noise, but we have tons of signal to noise with bold typically, and that's why everyone's using it. So that's a real factor with any of these techniques. You have to know your question. Is it really advantageous? Sometimes it's not. And also, I'm going to hopefully in the next episode try to talk a little bit more. And Faruk, you do this as well. Maybe we'll talk with layer fMRI. I mean, layer fMRI is potentially getting at things that Diana is trying to as well, and so with a little bit higher signal to noise and more robustness.
Very interesting, and I think it would be quite valuable to get different perspectives on this Diana paper, maybe from a statistics point of view, maybe from like other MRI experts' points of view, or electrophysiology researcher point of view. For instance, one thing that I was thinking when you were explaining the issues was that, well, we do select data in space. For instance, we select gray matter voxels to analyze, right? We don't want to go outside and select like an obviously cerebrospinal fluid voxel. So why should not we do like a similar thing in time? Maybe like that's what I was thinking as a counter argument of averaging everything, right? Then maybe we should average every voxel in the brain, but we know that this would, of course, like kill our signal. So I would be very, very curious as a listener to hear other perspectives.
Yeah, no, I agree with you. I agree. Like I said, it's a little bit tricky when you think about fMRI. When we put in any sort of model function, we're selecting for that, but we're mapping it. And if the map looked like noise, then we wouldn't be able to say much. But the key thing, I think, is to go from the time series to the maps, and have the maps speak for themselves. And even if you put in a model function, you clarify what it is. So right, I mean, it would be really good. I think that, and Russ talks about this a little bit. Other people have talked about this. That it's important to at least to clearly state everything that you've done, and try to use as objective measures as possible to do selecting, like independent measures, and then have like a show the maps or whatever. That we'll go from there.
Yeah. Let's keep some details for the future episodes, and let's see how the conversation will go. But
Yeah.
I think this would be in general, good idea for our podcast to maybe time to time discuss these hot papers, as we call, or hype papers, but also follow up in time and see how things are panning out, because as we know, like there are always different perspectives and different voices, different comments and opinions. I think it's valuable to communicate this aspect as well.
Yeah. Yeah.
Okay, so what else should we expect in this season? Like, do you have some other types of episodes that you have in mind? Like, we have people, of course, that are joining. They are talking about their own research and maybe how they came to their position. That also is very interesting as a listener, and their science, of course, they talk about. Then we started having this hot paper episodes, right? Sometimes we have multiple speakers discussing a topic. Do you have some other thing in your mind?
Yeah. Right. I mean, I think that there's a number of so like there's hot papers. There's some papers that come out. I kind of watch Twitter or whatever to see what's being discussed. I mean, there actually was there are a couple of interesting papers. I'm actually so like I mentioned before. I was intrigued by the idea of the challenge of parcellation, other things like types of resting state. I think it's one of these things that sort of keeps on coming up. Is like, okay, so you have things like quasi-periodic um, signals, global signal respiration. What's the purpose and what's in resting state. Uh, there's also interesting paper with Mike Breakspear and Alex Fernito were on it, in which they were using basically sort of a mathematical way, looking at the eigenspace of the brain structure and the eigenvalues of that brain structure sort of map out into where we map our functions, our gradients, and so there was some argument. There was that really published in a high impact journal that showed that hey, you can derive all the functional organization from just looking at the structure of this at the surface of the brain, and not even use necessarily the white matter or the function. And so really cool paper, but I think it got some pushback. I think it would be great to discuss that on the episode. But I think, yeah, that's an interesting concept. I mean, looking at deep evolutionary principles of where you tie this together in this sense. I mean, why is the brain organized in the way it is? I'd also like to even revisit. Even though I had 2 episodes with Jack Gallant, maybe I'll try to get some other people that were in his lab talking. I've always been intrigued with this idea. And there was another paper that came out recently of looking at language, saying, hey, it's not localized to language areas, it's everywhere.
Mm-hmm.
And this concept that's sort of emerging, especially pushed by Jack in some sense, where he had naturalistic stimuli, he put in these, multiple regressors modeling it, and making sort of this semantic map of the brain, showing that how you think of an object or a place or activity or anything is mapped everywhere depending on what it's associated with. And that's sort of like a model that's very different from our modular model, where we're looking at, this is the language area, this is the processing, and then it goes out. And so I'd like to talk a little bit more about that, to try to unite those two, or attempt to link those two ideas. A few other papers that I saw that sort of caught my attention. Every once in a while, I see a paper that fMRI is doing something that electrophysiology has kind of shown, and one thing that caught my attention was this idea of representational drift in activity, and the idea that if when you learn something, you have a pattern of activity that might be associated with it, but then over time, the neurons are always changing, and so that's drifting. Yet the representation stays the same. Like you're still able to recognize, a chair or a cup or whatever, but that representation of that in your brain is always changing. And so, there's a few fMRI papers. One that came out recently, I think it's still in bioRxiv, might be already out, but that sort of discusses this with fMRI. They study subjects over months, and they show this representational drift. And that links in directly with a pretty high impact paper that was out in Science, I think Science or Nature. So things like that, I always gravitate towards. Also like to do emerging areas, like there's a lot of work on focused ultrasound now,
Hmm.
various neural stimulation. I'd like to know where TMS is. Other techniques, there's near infrared light stimulation. So I'd like to get into other modalities a little bit more. I'm going to attempt to do that. Yeah, and along with that, I'd like to also maybe try to highlight not only OHBM but maybe other workshops or meetings. Now that things are opening up a lot more, almost completely, there's a lot of. I think lot of times people in my lab and other people I talk to, always say that OHBM is great and ISM is great, SFN is great, but it's these workshops that are really stimulating, and it might be worthwhile trying to get a highlight of those as well. And on top of that, I think people always like hearing about history. So I'm going to try to talk to more luminaries in the field to talk about deep history of either the deep history of OHBM, the deep history of some controversies, deep histories of cognitive neuroscience, and things like that. So I'm open to many ideas.
Yeah, I'm particularly interested in this part. I really like reading and hearing about the history of the ideas that we currently use in neuroimaging and neuroscience, because sometimes when you uncover the history, then all of a sudden things make more sense, or you are able to predict like what might come again in a few years time as an idea, as you were saying that this shape of the brain may be constraining the which functions that is doing. So I think that would be like a very nice kind of maybe a big picture episode.
Yeah, I agree. And keeping it, yeah, I mean, I think that it's hard to have a true debate. So it's always good to have sort of more of a conversation in which you sort of come to a consensus.
Yeah, exactly. Like having different perspectives, maybe at different episodes. I mean, we don't need to
Yeah.
have a debate, but we can have a debate over longer time by offering different perspectives to hopefully more people.
Yeah. Yeah. I completely agree with that. I completely agree. Um, and also, one of the things that I'm always trying to push as well is the more the nuts and bolts of clinical applications of fMRI and other neuroimaging techniques. It's such a problem. Uh, fMRI is so useful for the neuroscientists, and why can't it be more useful? And it's basically a problem of actually getting a foothold and having a platform that just works clinically. And because there's so many steps in creating the data, that it's not just sort of like an image that comes up that a radiologist looks at. So, and that's a real problem.
Mm-hmm.
That's a real challenge that we have to figure out how to overcome because there's so much latent potential in fMRI even now that could be used. So,
Yeah, indeed. Peter, do you have final things to say? I think I don't have more questions.
yeah, not really. I'm just looking forward to this is I'm having a lot of fun doing the podcast, and hopefully people I've been lucky in the sense that almost everyone I asked has agreed to come on, so that's great. And I've been fortunate to to luckily over the course of time to I think it's funny because if you're in the field long enough, you end up kind of knowing everyone, at least who's been in the field long enough. And I've been lucky enough to have gotten to know a lot of really amazing people from all over the world. And that's actually one thing I wanted to also emphasize is that I'm also trying to try to be more inclusive of interviewing people from underrepresented countries, underrepresented uh, you know uh, nationalities or whatever, and trying to emphasize that because I think that it's important to be mindful of that and to push that as much as possible. It's a lot of work, but it's a lot of fun.
Yes, it's a lot of fun and a lot of work at the same time, indeed. I'm also looking forward for the exciting new season.
Yeah, and also just a shout out to the producers of the podcast. It's yourself, Elfie Wern, who was the in charge of producing last
Yes,
season.
yeah.
Now it's you, and Jeff Mensch and Stefania Astanopoulos. It's not just me doing the interviewing, but there's a ton of work going on behind the scenes, editing, putting this up on what used to be Anchor now is Spotify for podcasters, and then on YouTube,
Yeah.
And also advertising it. So the four of you, or the producers in general, do a ton of work. So
yeah. Also, I would like to add that these are not the only people who, of course, contribute to our podcast or contributed last year. There are many others actually. Sometimes people come and go, and they edit like maybe 1 or 2 episodes and do other stuff. But I also would like to say that if someone wants to join to our efforts to contribute to the production of this Neurosalience podcast, this is a volunteer-based production. So it would always be nice to have some extra hands who can help us here and there. So maybe they can also email you. Can I say that?
yeah, please email me. Yeah.
Okay, great. Then, yeah, looking forward for the next season. Thanks, Peter.
All right. Well, thank you.
Neurosalience is brought to you by Organization for Human Brain Mapping. This episode is produced by Ömer Faruk Gülban and Alfie Wern.